So the next speaker is, forgive me if I get it wrong, Jehangir Cama.
Yes.
Who's from biological and biomedical physics, and is going to talk about optofluidic technologies for antibiotic testing and development. And it's all yours.
Thank you very much. As Rachel said, I'm Jehangir. I work with Professor Ulrich Kaiser in the Physics of Medicine labs down the road. And I want to describe the work we've been doing for antibiotic testing and development. Now, I think everyone here knows that antibiotic resistance is probably our most major public health crisis today. I mean, whenever I go back to Delhi in the last few trips, every time I've heard about someone else who's died because there's an infection which just can't be treated. So this problem is here. And their predictions, there was a report which came out, which was commissioned by the UK government, which predicts 10 million deaths annually due to resistant bacterial infections by the year 2050, with a massive cost, obviously, to the world economy. So we are really looking at going back to pre-penicillin days, which would be absolutely horrific, because most modern medicine depends on antibiotics, even for things like surgeries and chemotherapies as prophylactics. What are the major mechanisms of antibiotic resistance? So we can classify them into four basic areas. One is, if I get the pointer, that the cell barrier, the cell envelope, prevents the drug from crossing into the cell and reaching its targets. This can happen due to various resistant mutations in the barriers, in the cell envelopes, in the membranes. You can have this molecules coming into the cells, and then there are these efflux pumps in bacteria which act as vacuum cleaners and throw everything back out, and they can be overexpressed and you become resistant. Then you also have things like resistance mutations, where your target actually mutates and the drug doesn't work anymore. Or you can have enzymes created, if you've heard of these famous beta-lactamases. These are enzymes that are made by different bacteria which cut up beta-lactam antibiotics, and they are ineffective. Now, the major problem in this whole field actually is with the so-called gram-negative bacteria, because these actually have a double membrane cell envelope, which is extremely efficient at blocking the uptake of small molecule conventional antibiotics. And this is just a sort of schematic showing how complex this system is. But basically the issue is with these polymer brush-like, what are known as lipopolysaccharide molecules on the outer membrane of these bacteria. These form a very dense brush, which basically prevents these small molecules from going through. So molecular transport across this outer membrane and into the cells is basically governed by certain channels, protein nanopores, which sit and span the two membranes of the gram-negatives. So you can imagine that studying these transport processes becomes very important if we want to circumvent this delivery problem and get the drugs into the cells. So we need to understand molecular transport through these lipid membranes and through the protein nanopores in these membranes. So what we are developing is a two-pronged research strategy to look at this problem. The first thing is doing real biology, but with physics-based techniques. So we have these microfluidic chips, which are just little plastic devices which you bond to glass cover slips, and in them you have very small channels on the order of a few microns. And we can use this to confine bacteria. This is what's known as the mother machine, because what you have is a big channel for sending in your nutrients or your growth media, and little ladders on the sides, thousands of them, which are 1 micron wide. So you can fit your bacteria in single file, and the bacteria that gets seeded to the bottom, then when you start growing them, it starts dividing and going out as it fills up the channel. But that cell which has been seeded initially can't go anywhere, and that's the mother cell, hence mother machine. The important thing about using microfluidics and these techniques is that you have exemplary control over the nutrient microenvironment, because you have these syringe pumps and pressure pumps which are very well controlled. You know what nutrients you're dosing when you're doing your experiments. And this is a major problem with traditional biological experiments. They really don't keep good control over the nutrient microenvironment. And we have repurposed this technology to study drug uptake label-free using some UV autofluorescence microscopy. So as you can see, what we do is we use this chip, flow in our drug, and we realize that lots of these antibiotics, if you shine a little bit of UV light on them, they are autofluorescent, so you can track them label-free. And all we are doing is studying how that accumulates in these single bacteria, and we can do this with hundreds of cells for now, and we are scaling up the chips so we can do them for thousands of cells. But the important thing is, as I said, you control the nutrient microenvironment. So we can actually study the role of different nutrients, different pH levels, and specific environmental signals in drug uptake. And this is just some representative traces where we can actually study these dose response curves in individual cells. And we have a sort of general permeability model, which is already showing us differences between bacteria that are dormant in stationary phase or bacteria that have been growing for a few hours and in their exponential phase of growth. But why do we really need single cell resolution as well? Because it is quite well known that due to the inherent stochasticity in gene and protein expression, even within genetically identical populations of cells, you have, say, 1% or 0.5% of your cells that tolerate antibiotics. No one quite knows why. These are known as persister cells. There are other subphenotypes known as viable but non-culturable cells, which really can't be studied with conventional techniques. Now, if you look at this video, you see that initially all these cells took up the antibiotic, but slowly you'll see a few which are just expelling them. And these are very likely candidates for persisters because they've thrown out the drug and are probably going to survive the antibiotic dose. So we are starting to see hints of these phenotypes in our experiments, and we can then start tagging certain genes which we suspect are involved in persistence and getting biomarkers for these phenotypes. But I said that we have a nice model to work out the permeability in the whole cell. The problem is that the targets of these antibiotics typically are in the cytoplasm inside. So from these whole cell measurements, we need to apply various models, and I'm not going to go into the details of this, but what we want to know is how much of the drug is stuck in the outer membrane, how much is in the periplasm, and how much is actually reaching the cytoplasm and the drug targets. And we're working, this is just work in progress at the moment. We've got some collaborations in Exeter with people actually modeling these processes, and what we're doing at the moment is looking at different knockout strains. We've done all this work with E. coli, but the same techniques can be used for other gram-negative bacteria. And when you knock out, say, a channel, an efflux pump, the shape of this curve changes. When you knock out one of the important protein channels, it changes. So we want to understand how these specific transport systems affect the accumulation of the drug inside the cell. But that model is extremely complicated. Biology is messy. There are lots of parameters. And this brings me to the complementary techniques. We really need reductionist biophysical approaches to complement these whole cell measurements to get more specifics of these processes. And this is work we've been doing for now a couple of years, where we reduce the problem. We are physicists, right? So we are reductionists. We take what are known as liposomes as model systems. This is simply a lipid bilayer which mimics the outer membrane of a bacterium or a cell. And these are very nice because it's fully controlled. You make the membrane yourself, and we reduce the problem to simply diffusion across a membrane. And these are also nice cell mimics. They can be done with conventional microscopy. And again, we're using our UV autofluorescent drugs to study these transport processes. And we only study the process, the part of the cell important to transport, which is the membrane. So the system is very simple. We have a T-junction microfluidic device where we feed the vesicles in from one side, you feed your drug in from the other side, you mix the two, and as these vesicles go through this serpentine sort of network, all you have to do is video microscopy at different time points, and you can sort of see over here the vesicles in the beginning when they've just been mixed are dark, and as they flow through the system, they become lighter and lighter as the drug molecules move into them. So that's the basic idea behind this technique, and we've used it to do quite a few things. We've looked at the pH dependence of certain drugs and their transport rates. Why is pH important? Because lots of these molecules have multiple protonation sites, so depending on the pH of your system, they have different charges. And if you have something that's charged, it's not going to like to sit inside the hydrophobic core of your lipid bilayers, and therefore its transport rates go down. We've looked at the role of different types of lipids and found quite interesting things that even lipids from the same sort of family can show orders of magnitude differences in drug permeability, and this is due to the specifics of the molecular composition of the membrane. But what we've also managed to do is isolate and purify proteins of interest from the outer membranes of gram-negatives. In this case, it was the outer membrane protein F from E. coli, reconstitute it in these vesicles to make what are known as proteoliposomes, and do the same sort of assays. And now what we can do is measure drug transport across the pure lipid bilayer, and then we can measure drug transport across that same lipid bilayer, but with these proteins stuck inside it. And by subtracting the two contributions to drug permeability, we can work out the transport rates through these proteins in a nice controlled system. So at the moment, we used to have lots of problems with these vesicle-based systems because ways of making them were messy. And lipids are difficult to work with. But over the last year, we've spent a lot of time establishing this new microfluidic way of making lipid vesicles in our lab. This is a 6-way junction where you have your lipids dissolved. They always have to be in an organic phase. We have octanol over here, so the lipids are dissolved in octanol coming through here. You have 2 aqueous phases, the outer aqueous phase and the inner aqueous phase, and you're basically blowing bubbles. This is just basically a fluorescent moiety for visualization. And what you have then is a little droplet of octanol which buds off due to interfacial energy minimization. And you then have pure lipid vesicles or liposomes, and your octanol can be separated by a density separation because it is lighter than water, so that's fine. And then this circumvents lots of issues that we had with our earlier vesicle formation technique. It's much higher throughput. It's monodisperse. You can see there's very nice size distribution over here. Importantly, we can use physiological salts, which we could not with the old techniques. Encapsulation is much better. It's much more scalable. And by mixing proteins into the lipid octanol phase, we've homogenized the lipid to protein ratios at source. And we've shown that we can also have nice uniform incorporation of these sort of boards, these proteins in these liposome membranes. The other thing you can do when you have all this on chip is that you can either take it into, say, a T-junction and do the same sort of flow assays that I showed you earlier, or we've also made these sort of trap arrays. So we've got multiple parallel arrays of traps where you can hold these liposomes for hours or even days and do long-term experiments on them. So we can study drug transport processes which are slower than could be measured earlier. But we are also using this platform with one of our sponsors here, NPL, to study the activity of new types of antibiotics which directly target and lyse bacterial membranes. These are polypeptide-based antibiotics which work, you need to study these things over a couple of hours. But importantly, using these sort of systems, you can do a very important safety test as well. Because if you have a drug which rips apart lipid membranes, you want to make sure that if you inject it into yourself, all your red blood cells don't lyse and you have hemolysis. So with this system, you can actually make mimics of mammalian membranes and bacterial membranes and do the same tests with these drugs to make sure that they are selective only for the bacterial mimic membranes. So in summary, I hope I've shown you that we've got a suite of techniques now looking at antibiotic development for the problem of antibiotic resistance and failure in gram-negatives. We are combining both whole cell approaches and biomimetic approaches to get a comprehensive overview of drug transport in these gram-negative systems. And besides fluorescence, we are developing alternative drug detection techniques such as absorbance. But one question I would really like to put to this audience is if you have any other ideas for label-free detection of drug molecules, please come and talk to me. We are looking for them. And our biomimetic system with these vesicle traps is also being used for quantifying polypeptide antimicrobial efficacy with our collaborators at NPL for new drug development. Finally, I need to acknowledge a lot of people, Ulrich, in whose group I've worked for many years, and I'm very happy over there, and we've really come quite a way since I started my PhD, I think. Michael and Karim are 2 excellent PhD students who are doing all the work with the OLA system, with the microfluidic vesicle formation system. Stefano is the person with whom we've been doing the work on the mother machine. He is now based at Exeter, but was previously a Cambridge person. Matthias Winterhalter has worked with us on the porins and the OMPEF proteins. He is based in Bremen. Max, as I said, for polypeptides is at NPL. Krasi for modeling. Case Decker invented the OLA liposome formation technique and very helpfully showed us how to make it and make it work. The rest of our group and the biological and soft systems community, and of course the Cavendish. Thank you very much.
Thanks very much, Jehangir. I almost forgot I wasn't a biologist listening to that. That was very good. Questions, quickly. Everyone else isn't a biologist, no.
There are some biologists in the Cavendish. Yes.
Do we have any? We do have time. No, I guess they're blown away. So thank you very much.
Thank you very much.